Bioinorganic chemistry is a field that examines the role of metals in biology. Bioinorganic chemistry includes the study of both natural phenomena, such as behavior of metalloproteins, as well as artificially introduced metals, including those that are non-essential in medicine and in toxicology. Many biological processes, such as respiration, depend upon molecules that fall within the realm of inorganic chemistry. As a mixture of biochemistry and inorganic chemistry, bioinorganic chemistry is important in elucidating the implication of electron transfer protein, substrate binding and activation, atom and group transfer chemistry, as well as metal properties in biological chemistry. The successful development of truly interdisciplinary work is necessary for the advancement of bioinorganic chemistry. As the composition of all life includes carbon, nitrogen, calcium, sodium, chlorine, potassium, hydrogen, phosphorus, oxygen, and sulfur. The organic compounds, that is protein, lipids, and carbohydrate, contain the majority of carbon and nitrogen. And most of the oxygen and hydrogen are present in water. The entire collection of metals containing biomolecules in a cell is called as metallome. A diverse collection of transporters, which may be in the form of vacuoles, storage protein, and small molecules, are employed to control metal ion concentration and bioavailability in living organisms. Crucially, many essential metals are not readily accessible to downstream protein, owing to low solubility in aqueous solution or scarcity in the cellular environment. Hence, metal containing electron transfer protein are also common. They can be organized into three major classes: iron sulfur protein, copper protein, and cytochromes. These electron transfer proteins are complementary to the non-metal electron transporters, such as NAD and FAD. The nitrogen cycle makes extensive use of metals for the redox interconversions. Iron sulfur protein are proteins characterized with the presence of iron sulfur clusters containing sulfide-linked di, tri, and tetra iron centers in variable oxidation states. Iron sulfur clusters are found in a variety of metalloproteins, such as ferredoxin, as well as NADH dehydrogenase, hydrogenase, coenzyme Q cytochrome C reductase, succinate coenzyme Q reductase, and nitrogenase. Iron sulfur clusters are best known for their role in oxidation reduction reactions of electron transport in mitochondria as well as in chloroplasts. Both the complex 1 and complex 2 of oxidative phosphorylation have multiple iron sulfur clusters. Additionally, some iron sulfur protein regulate gene expression. Iron sulfur protein are vulnerable to attack by nitric oxide, forming dinitrosyl iron complexes. In most iron sulfur proteins, the terminal ligands on iron are thiolate, but exceptions are also there. These proteins affect the metabolic pathway of most organisms. In most of the iron sulfur proteins, the iron centers are tetrahedral and the terminal ligands are thiolate to sulfur centers from cysteine residues. The sulfide group are either 2 or 3 coordinated. The distinct kind of iron sulfur clusters with these features are most common. There may be 2 Fe2S clusters, as is present in ferredoxin, 4 Fe4S clusters, as is present in aconitase, 3 Fe4S clusters, etc. A discussion on 2 Fe2S cluster. The simplest polymetallic system, that is Fe2S2 cluster, is constituted by 2 iron ions, bridged by 2 sulfide ions, and coordinated by 4 cysteine ligands. In Fe2S2 ferredoxin, it is flanked by 2 cysteines and 2 histidines. The oxidized protein contain iron in the ferric state, whereas the reduced protein contain iron in the form of ferrous ion. These species exist in 2 oxidation states, that is Fe positive 3, Fe positive 3, and Fe positive 3, Fe positive 2. Iron sulfur domain is also associated with 2 Fe2S clusters. On to 4 Fe4S cluster. There are 4 iron ions and 4 sulfide ions placed at the vertices of cubane type cluster. The Fe centers are typically further coordinated by cysteine ligands. The Fe4S4 electron transfer protein, that is Fe4S4 ferredoxin, may be further subdivided into low potential and high potential ferredoxins. Low and high potential ferredoxins are related by the above redox scheme. Some 4Fe4S clusters bind substrate and are thus classified as enzyme cofactors. For example, in aconitase, the iron sulfur cluster binds aconitate at one iron center that lacks a thiolate ligand. The cluster does not undergo redox, but serves as a Lewis acid and converts citrate to isocitrate. In 3Fe4S cluster, there is one iron less than Fe4S4 core. 3 sulfide ions bridge 2 iron ions each, while the 4th sulfide bridge 3 iron ions. The formal oxidation state may vary from Fe3S4 positive, where all the iron is in the ferric state, as well as in the other form, where all the iron is in the form of ferrous state. In a number of iron sulfur protein, Fe4S4 cluster can be reversibly converted by oxidation into Fe3S4 cluster. Then, for example, the inactive form of aconitase possesses Fe3S4 cluster, where the iron is in the form of Fe positive 2. There may be several other types of iron sulfur clusters. A more complex polymetallic system is commonly found in nitrogenase, carbon monoxide dehydrogenase, and hydrogenase. There is a special 616 coordinated cluster found in hydrogenases. Nitrogenases are enzymes that are produced by certain bacteria such as cyanobacteria and rhizobacteria. These enzymes are responsible for reduction of nitrogen into ammonia, that is during nitrogen fixation. Nitrogenases are the enzyme commonly found in rhizobacteria and cyanobacteria. The enzyme catalyzes the conversion of nitrogen into ammonia, which is the key step in the process of nitrogen fixation. Nitrogen fixation is required for all forms of life because nitrogen being essential for the biosynthesis of molecules such as nucleotides and amino acids. A typical nitrogenase complex is composed of two parts, a reductase and a nitrogenase. Reductase is an iron protein, that is a homodimeric iron protein, which has a high reducing power and is responsible for supplying electrons onto the nitrogenase part. Nitrogenase is a molybdenum iron protein. It's a heterotetrameric protein, which uses the electrons supplied by the reductase and uses these electrons to convert nitrogen into ammonia. The reductase part, that is iron protein, it's a dimer of identical subunits, which contains one Fe4S4 cluster. The function of Fe protein is to supply electrons from a reducing agent such as ferredoxin or flavodoxin onto the nitrogenase protein. The transfer of electrons requires an input of chemical energy, which comes by the binding of ATP. ATP hydrolyzes and supplies energy for the transfer of electrons onto the nitrogenase part. Hydrolysis of ATP also causes conformational changes within nitrogenase complex. Binding of iron protein and MOFE protein closer together for easy electron transfer. The MOFE protein or the nitrogenase part is a heterotetramer consisting of 2 alpha subunits and 2 beta subunits. The MOFE protein also contains 2 iron sulfur clusters known as P clusters, which are located at the interface between the alpha and beta subunits, and 2 FeMO cofactors within the alpha subunit. The oxidation state of MO in these nitrogenases was earlier thought to be MO, but recently it is thought to be in the +3 oxidation state. Molybdenum in other enzymes is generally bound to molybdopterin. The core Fe8S7 of P cluster takes the form of 2 cubes, that is Fe4S3, which are linked by a central carbon atom. Each P cluster is covalently linked to MOFE protein by 6 cysteine residues. Each FeMO cofactor, that is Fe7MOS9C, contains 2 identical clusters of Fe4S3 and MOFE3S3, which are linked by 3 sulfide ions. Each FeMO cofactor is covalently linked to alpha subunit of the protein by 1 cysteine residue and one histidine residue. Electrons from iron protein, that is the blue part, that is also known as reductase, enters MOFE protein or the nitrogenase shown in purple at the P cluster, which then transfers the electron to FeMO cofactor. Each FeMO cofactor acts as the site for nitrogen fixation, with the nitrogen binding in the central cavity of the cofactor. Thus, nitrogenase complex is a nitrogen fixing, is an enzyme which is involved in fixation of nitrogen. Its main role is to convert nitrogen into ammonia and thereby making it available for the bioorganisms. This nitrogenase enzyme is found in nitrifying bacteria, that is rhizobacteria or cyanobacteria. Higher organisms are unable to convert nitrogen into ammonia. Hence, they require the help of nitrifying bacteria, which are able to convert nitrogen into ammonia. Next is cytochrome. This is again a redox protein which contains a heme with iron as cofactor. It may be classified into cytochrome A, cytochrome B, cytochrome C, and cytochrome D. Cytochrome B is found within both molecular and cell biology. It's an important protein found in the mitochondria of eukaryotic cells. It functions as electron transport chain and is the main subunit of transmembrane cytochrome BC and cytochrome BF6 complexes. Cytochrome B is a component of respiratory chain and is also known as cytochrome BC complex or ubiquinol cytochrome C reductase. In plants, it's found in the chloroplast in the form of cytochrome B6 and is involved in electron transport. During the pumping of proton, it creates proton motive force. A proton gradient is used for generation of ATP. Regarding its structure, cytochrome B is an integral membrane protein. It consists of 2 heme groups. 4 conserved histidine residues are postulated to be the ligands of the iron atom of these 2 heme groups. Cytochrome C is another electron transfer protein. It is used to catalyze the chemical reactions involving redox processes. Cytochrome C or heme-containing protein have heme carbon covalently attached to the peptide backbone via thioether bond. These bonds, in most cases, are specifically in the form of cysteine, X-cysteine, and histidine. Binding motive where X can be an amino acid. Cytochrome is another heme protein, another protein containing a heme group as a cofactor. Cytochrome D, as other proteins of its family, is a membrane-bound heme protein. But unlike cytochrome A and B, cytochrome D has heme D instead of heme A or heme B. Cytochrome D is a triheme oxidase. Its main function is the reduction of oxygen into water. It has an active site with iron instead of copper and needs no proton pump as they can produce a proton motive force themselves. This was all regarding nitrogenase and cytochromes.